Welcome back, everyone, to the deep dive. For this deep dive, we're going to go into the world of Leptospira
Oh, cool.
from a medical microbiology perspective.
All right.
We'll be referencing the book, Principles and Practice of Clinical Bacteriology.
Sounds good.
We'll be looking for those little clinical pearls or just things that are interesting to us, as medical microbiologists.
Awesome.
So to get started, why don't you tell us a little bit about what Leptospira is?
So Leptospira is a spirochete,
Oh, okay.
and so it's going to be that really thin, tightly coiled
Mhm.
spiral shape. Sometimes they even have hooks at the end,
Oh, wow.
which can be interesting, especially when you're looking under a microscope.
So they're really hard to see, I imagine.
Yeah, they are really difficult to see. They're also gram-negative,
Okay.
and as you know, gram-negative organisms can be kind of finicky with staining.
Right.
They're also slow-growing,
Mm.
and they need special media like EMJH,
Okay.
which is enriched with vitamins and fatty acids.
Interesting.
Yeah, it's like they need like a 5-star meal to grow.
That's funny. Now, I remember back when I was training, we only learned about 2 different species of Leptospira.
Right.
There was Leptospira interrogans and Leptospira biflexa,
Mm-hmm.
and they were just either pathogenic or non-pathogenic.
Yeah.
But in this book, Principles and Practice of Clinical Bacteriology, they seem to indicate that there's been an update to that classification system.
Yes, we don't really use that system anymore. That's kind of a relic of the past.
Oh, wow.
Now we use genomics to help us classify these organisms.
Okay.
And with genomics, we've discovered that there's at least 16 different species.
Wow.
Yeah. And we've also discovered that pathogenic and non-pathogenic reservoirs can exist within the same species.
Okay, so it's more complicated than just the species.
Exactly, yeah. So that old system really wasn't able to capture the true diversity of all
Yes.
these different Leptospiras.
So how are we identifying these species now?
So we're using 16S RNA gene sequencing to identify the species.
Okay.
But then for further classification, we still use serological methods. Especially when we're trying to figure out the over 230 serovars
Wow.
organized into 23 serogroups.
That's a lot.
It is a lot to keep track of.
Yeah, for sure. So the book also mentions that leptospirosis is actually one of the most widespread zoonoses globally.
Yeah.
Like, it really is a true globetrotter. It's on every single continent except for Antarctica.
That's right.
Why is that?
Well, Leptospira thrive in warm climates and damp soil.
Okay.
And so anywhere where you're going to have a lot of animal interaction in those conditions is
Yeah.
going to be a good place for them to thrive,
So basically anywhere that's not Antarctica.
pretty much.
All right, that makes sense. So in the book, they also mentioned these terms called maintenance hosts and accidental hosts.
Right.
Can you explain that to us?
Yeah, so maintenance hosts are the animals that are the primary reservoirs for Leptospira.
Okay.
And these are usually going to be rodents and small mammals.
Okay.
And they carry the bacteria without showing any symptoms.
Oh, wow.
And they can shed the bacteria in their urine contaminating water and soil. Okay. And then accidental hosts, like us humans, get infected through contact with that contaminated water or soil.
That makes sense.
Yeah. So these rodents are kind of going around silently spreading the bacteria.
Yeah, you don't really know where it is.
Exactly. And that's what makes it so difficult sometimes.
Yeah, and how do these bacteria actually cause disease?
So Leptospira enters the body through cuts or abrasions in the skin,
Huh.
or even through mucous membranes. And then it travels to the bloodstream.
Hmm.
And then from there, it can disseminate to various organs like the liver, kidneys, and the central nervous
It can
system.
go all over.
Yeah, it really can.
And what kind of symptoms would we see?
So the clinical manifestations can vary widely.
Okay.
It can range from a mild flu-like illness
Hmm.
to severe and life-threatening disease.
So it can be really mild or really severe.
Exactly. And that's what makes it so challenging from a clinical perspective.
Yeah, you never know what you're going to get. And then on top of that, you mentioned the book says that there's this unique biphasic nature to the disease.
Yes. So there's an initial phase called the leptospiremic phase,
Okay.
where the bacteria are circulating in the bloodstream.
Okay.
And this usually lasts for about a week.
All right.
And then after that, we have the immune phase,
Mhm.
where the body is mounting an immune response and producing antibodies to fight off the infection.
So it's like 2 different phases of the disease almost.
Exactly.
And then the book mentions laboratory diagnosis as being crucial
Absolutely.
because of all this variation.
Right. Right.
So to start talking about that, why don't we dive into some of the diagnostic techniques for identifying Leptospira?
Okay, so we'll start with the classic microscopy.
Okay.
But as we mentioned before, Leptospira can be really difficult to see under the microscope because they're so thin. And those traditional staining methods aren't always reliable.
Okay.
You could try dark field microscopy.
Okay.
That might help you visualize those characteristic spirals. But we have to be careful not to mistake any artifacts for Leptospira.
That makes sense. So is there anything else besides just straight microscopy?
So we can also look for them in tissue samples using histopathology.
Okay.
Silver staining techniques like the Warthen starry stain can highlight spirochetes, but it's not specific for Leptospira.
Okay.
So other spirochetes could look similar under the microscope.
Gotcha. So what about using antibodies?
So we could use immunohistochemistry, which uses antibodies specific to Leptospira to help visualize them in the tissue sections.
And that would be more specific?
Yes, a lot more specific.
Awesome. What about culturing it? Can we culture a Leptospira?
We can, but it's not easy.
Okay.
They're very slow growing, and as we mentioned before, they need that specialized media.
Right, the EMJH.
Yes, EMJH, exactly.
Like a 5-star meal.
Exactly.
So what kind of samples would we try to culture?
So during that initial leptospiremic phase, blood cultures are best.
Okay.
But once we're past that first week, then urine is a better sample.
Okay, so blood in the first week and then urine after that.
That's right.
Okay, and then the book really emphasized the importance of specimen handling.
Yeah, because Leptospira is fragile.
Okay.
So urine samples need to be neutralized and centrifuged to concentrate the bacteria before we attempt to culture them.
It sounds really complex.
It is. It definitely takes some expertise.
And once you culture it, then you still have to identify it.
Yes, exactly. We would use molecular or serological techniques to identify the specific serovar.
Okay, so what are some of those molecular typing techniques that we can use?
Well, pulsed field gel electrophoresis, or PFGE, is the gold standard for a definitive identification.
Okay.
It analyzes the entire genome, so it allows us to differentiate between serovars with incredible precision.
Wow, so it's really specific.
Yes, very specific.
What about PCR?
Yeah, PCR is faster and more sensitive than traditional culture methods.
Okay.
And the book mentions some PCR-based techniques that are especially helpful for Leptospira, like PCR-REA and LSSP PCR, and these can be used directly on clinical samples.
Nice. So you don't have to culture it first.
Exactly. So we can potentially diagnose leptospirosis within hours rather than days or weeks.
That's really helpful.
Yes, it is.
All right, so we've talked about some molecular methods. What about serology?
So for serology, the gold standard test is the microscopic agglutination test, or MMET.
Oh, yeah, the MMET.
That's right.
So how does that work?
So for that test, we mix the patient's serum, which would contain the antibodies, with live Leptospira antigens. If the patient has antibodies against Leptospira, they will bind to the antigens and cause the bacteria to clump together. And that's called agglutination. And we look at this under a microscope. And the amount of agglutination that we see can help us determine whether or not the patient has been infected.
Okay, that makes sense. I remember learning about this test in school, and I remember cross-reactivity being a big issue.
Yes, cross-reactivity is a big issue because Leptospira serogroups can share some common antigens. And so the antibodies produced against one serogroup could react with another. And so it can be hard to pinpoint the exact infecting serogroup.
Especially early on.
Yes, especially in the beginning.
So basically, if it's positive, it doesn't really tell us a whole lot.
Not exactly, no. It just means they have antibodies against Leptospira.
Gotcha.
But to add to that complexity, we have something called the paradoxical immune response. The paradoxical immune response.
What is that?
So sometimes the strongest immune response or the highest antibody titers are directed against a serogroup that's not even causing the infection.
Oh, wow. That's interesting.
Yeah, it's like the immune system is playing a trick on us.
Yeah, that makes it really hard to interpret.
Right, it really does.
So what do we do?
So we have to consider the patient's presentation, the timing of sample collection, and the patterns of cross-reactivity. And we may need to test multiple samples over time to see how the antibody titers are changing.
Okay, so it's like a puzzle. You got to put all the pieces together.
Exactly. It's a lot like solving a puzzle.
That's good. Is the MAP the only test that we can do?
No, we also have ELISA and IgM dipstick assays. These are faster and easier to perform, and some are even commercially available. So that makes them a lot more accessible to labs that may have limited resources.
Gotcha. So we have a whole bunch of different tools that we can use to identify Leptospira.
Yes, and we have to pick the right tool for the job.
Exactly. All right, so let's move on to treatment.
Okay.
What are the options for treating Leptospira infections?
So it really depends on the severity.
Okay.
For mild cases, supportive care is usually enough.
Okay.
So that would just be things like rest fluids, over-the-counter medications.
Okay.
But for severe cases, then we need to ramp it up a little bit.
Okay, so like hospitalization?
Yeah, hospitalization, intensive care, close monitoring.
Yeah, all that stuff.
Exactly.
So what about antibiotics?
That's the big question, isn't it?
It is.
There's some debate surrounding antibiotic use, but we think that early intervention is key.
Okay.
Antibiotics like doxycycline and penicillin has shown some promise in shortening the illness and maybe even preventing complications, but that's only if they're given early.
So that first week, while it's still in the blood.
Exactly, before it has a chance to invade the tissues.
Gotcha.
Once it has invaded the tissues, antibiotics become a lot less effective.
So it's not really a magic bullet.
Not really. No.
But it can be helpful.
Right.
And we shouldn't forget about those supportive measures as well.
Yeah, those are really important, especially for the severe cases.
Okay. All right, so now let's talk about the bigger picture.
Okay.
How do we control and prevent Leptospira infections?
So this is where the One Health concept comes in.
Okay.
We have to recognize that human, animal, and environmental health are all interconnected. So controlling those animal reservoirs, particularly rodents, is going to be really important.
So basically rat control.
Yes, rat control.
Gotcha.
We also want to think about vaccinating livestock and domestic animals, to try and reduce the spread.
And then what about people who are working in high-risk occupations?
So for them, we want to encourage them to wear personal protective equipment.
Okay.
And we can also consider chemoprophylaxis with doxycycline.
Okay. What about vaccines for humans?
So there are some, but they're not widely used.
Okay.
There's a trivalent vaccine that's used in Cuba,
Okay.
but its availability is limited. And we still have some challenges with current animal vaccines.
Okay.
They may not prevent the bacteria from colonizing the kidneys and being shed in urine.
Oh, wow.
So even vaccinated animals could still spread the bacteria.
Oh, so there's still more work to be done.
Yes, there definitely is.
To create better vaccines.
Yes, better vaccines for both animals and humans.
Okay. That's great.
Yeah.
I think this has been a really great overview of Leptospira.
I agree.
Now, I think to test your knowledge,
Okay.
I have a few exam questions for you.
Sounds good.
So first question,
Yeah.
True or false? Serological testing alone is enough for a definitive identification of the infecting Leptospira serovar.
What do you think?
Take your time.
I'm thinking.
All right, time's up.
Right.
The answer is false.
No.
Serological tests like the MA are great
Right.
for detecting Leptospira antibodies, but they can't pinpoint the exact serovar.
That's right. You need to isolate the organism to do that.
Exactly. All right, next one, question 2.
Okay.
Which of the following is not a common feature of severe leptospirosis?
Okay.
Your options are A, acute renal failure, B, pulmonary hemorrhage, C, pancreatitis, or D, cardiac arrhythmias.
All right, let me think about this.
Take your time.
Okay, I think I got it.
All right, the answer is EC, pancreatitis.
Okay.
So while Leptospira infection can elevate amylase levels, pancreatitis itself isn't usually seen.
That makes sense.
Okay, question 3.
Okay.
The microscopic agglutination test, or the MAT,
Yes.
is the gold standard for serological diagnosis of leptospirosis. What is its major limitation?
Ooh.
Think back to what we were just talking about.
I think I know.
The answer is cross-reactivity between serogroups.
Yes.
This can really complicate the interpretation of the results.
Especially in those acute phase samples.
Exactly. All right, question 4.
Okay.
This one's a little more open-ended.
All right.
Describe the role of rodents and small mammals in the life cycle of Leptospira.
Okay. So rodents and small mammals are the main maintenance hosts for Leptospira.
Exactly.
They carry the bacteria without showing symptoms and shed it in their urine, contaminating water and soil.
And then we get it.
Exactly.
All right, last question.
Okay.
Question 5. Discuss the rationale and challenges associated with antibiotic use in the management of leptospirosis.
Oh.
This one brings a few things together.
Right.
Think about when we would want to give antibiotics.
Yeah.
And what are the good things about them and the bad things.
Okay, I think I got it.
All right, so we use antibiotics to try and shorten the illness,
Right.
reduce the severity of the symptoms, and prevent complications.
Mm-hmm.
And doxycycline and penicillin are the usual choices.
Okay.
But the challenge is timing.
Right.
They work best early on before Leptospira invades the tissues. But even then, the evidence for them working in severe cases is mixed.
Yeah.
So we really have to consider the risks and benefits for each patient.
Exactly.
And we can't forget about supportive care.
Yeah, supportive care is very important.
All right, so that wraps up our exam questions.
That was fun.
Glad you enjoyed it.
I did.
This deep dive was just a little glimpse into the world of Leptospira.
It was.
And there's still so much more to learn.
Oh, yeah.
But I think this was a really great overview.
Yeah, it was a great starting point.
We encourage you to go check out those source materials.
Yeah, definitely.
And keep learning.
And until next time, keep those lab coats on.